Hi everyone. Now, we're going to discuss about Chapter 6, Expression of Biological Information. There are 4 subtopics in these chapter: 6.1, DNA and Genetic Information, 6.2, DNA Replication, 6.3, Protein Synthesis, Transcription and Translation, 6.4, Gene Regulation and Expression, lac operon. Before we can continue, let's look up into the learning outcome for 6.1. At the end of this video, students should be able to state the concept of central dogma. Let's us start. Let's us look at the concept of central dogma. Central dogma concept was named by Francis Crick in 1956. Cells are governed by a molecular chain of command with a directional flow of genetic information shown below. DNA to RNA to protein then trait. What is DNA? DNA stay in the nucleus. DNA can copies itself and gets passed on to new cells. DNA can replicate itself through process of DNA replication. DNA also holds instructions for making protein. DNA undergo process of transcription to synthesis RNA. RNA is made in the nucleus and goes to the cytoplasm. RNA undergo translation process to synthesis proteins. Protein are used to make body structures such as muscle, bones, skin and hair. Protein are also made up enzymes, transporters and hormones in the body. While trait is an observable characteristic that determine who you are. As for the concept of central dogma of molecular biology, information passes in one direction from DNA to RNA copy of the gene and directs the sequential assembly of a chain of amino acids into a protein. Central dogma of molecular biology provides an intellectual framework that describes information flow in biological systems. Now we are going to discuss about DNA replication. Watson and Crick proposed that DNA replicates by semi-conservative model. These model proposed that base pairing allows each DNA strand to serve as a template for a new strand. Semi-conservative replication of DNA involves 2 parental DNA strands that later separate each other. Each of those strands then serves as a template for the synthesis of a new DNA strand. As for the result, 2 molecules of DNA double helix form, both of which consist of 1 parental and 1 new strand. Let's watch this video to increase your understanding.
Let's talk about semi-conservative DNA replication. What I'm looking at here is a DNA double helix. And one thing to keep in mind when I'm looking at this DNA double helix is it's just a little cartoon of a tiny piece of DNA. In an actual cell, a chromosome's DNA can contain millions of base pairs. So if I were to draw out this whole thing, it would have to wrap around this room many, many, many times. But looking at a small piece makes the cartoon easier. So I'm looking at a DNA double helix before replication. So I'm looking at the old DNA before it's replicated. And semi-conservative DNA replication tells us that this strand here is going to be a template for building its complementary strand, and this strand here is going to be a template for building its complementary strand. So because of complementary base pairing, this can be used to build its partner, its complementary strand. And also this strand here can be used as a template to build its partner, its complementary strand. Giving me 2 daughter DNA double helices, where here in pink I've showed the new DNA. This process here is DNA replication. And we have another cartoon for looking at this when we're studying the details of how the cell does this, where we're looking at a DNA double helix that's been opened up to form a replication bubble so that the cell can replicate its DNA. So I've looked here shortly after it started. So it opened here at the origin of replication, and enzymes have built the new DNA, or have started to build the new DNA, really. And I'm not going to go into too much detail about how this is built, but kind of keep in mind that what this is a cartoon of the cell building this new DNA. So the blue template DNA is the same in both of these cartoons, and the pink new DNA is the same in both of these cartoons. So that when the cell is done, they'll have down here one new DNA strand, one DNA double helix that has an old strand and a new strand, and up here it'll have a DNA double helix with an old strand and a new strand. And these are the same strands that we're looking at here.
Please scan QR code below to answer the questions, or you can access the questions via given link. Thank you for watching and bye-bye.